All right, as we just wrapped up mitosis, let's take a look at how that's similar that it is into meiosis. Again, we start at interphase. Interphase, again, has those same parts. We have the G1, S, and G2. Those are all going to be involved in either one of those, where we have the first growth, DNA is going to be copied, and then the organelles are going to be copied. From there, now we're going to go into another M phase, but instead of division of the nucleus of mitosis of a body cell, now we start to divide sex cells, and in particular, these are called gametes. So this process is known as meiosis. So we go through this process here, we go to M1, which is meiosis 1, then it will go through a whole process again, M2, and then we'll end with the cytokinesis again, ending with our 4 cells that we saw in the first video. Now, we kind of draw those out. We start off with our 46 chromosomes, and then we go to our 92. So if we had our DNA in a nucleus all jumbled up in here, by the way, one of the terms that we use for this jumbled up DNA is called chromatin. Chromatin. And once it starts to get condensed up, it's going to make the chromosomes. So let's work our way into meiosis 1. Meiosis 1 is a very important step. Highlighted star, do what you need to. This is prophase 1. And in prophase 1, we're going to have a few things take place. One of the things we want to remember a little bit before we get into it is how chromosomes are arranged. Now remember, just draw a quick thing. It's called a karyotype. We have all of our chromosomes mapped out. We should have 23 pairs of them, and the 23rd pair being our XX or XY, depending for male or female. But if I just look at chromosome set number 1, I really want to zoom in on that set. What we're going to have on that set, we're going to have one chromosome from mom, one chromosome from dad, coding, each coming one from a sperm, one from an egg. Now, in the S part of interphase, we're getting its identical copy. Here is its identical copy, held together in the middle by the centromere. Now, what I want to do is, in prophase 1, we want these guys to get together. These two pairs are homologous. They are similar. The coding for chromosome 1, we'd have the same thing chromosome 2, chromosome 3, all the way up to the 22 autosomes and the 23rd sex chromosome. So let's zoom back out of that, back up to the forefront. So in prophase 1, we have our cell, and the same thing occurs in mitosis is that nucleus has to dissolve. The chromosome structures find each other and condensed up. Okay? Now, I've drawn a total of 4 chromosomes, or I should say 8 chromosomes, 1, 2, 3, 4, 5, 6, 7, 8. But if I were to really zoom in on it, it's going to basically looking at just chromosomes that's 1 and 2. Okay? Now, the next thing that's going to happen in prophase 1 is that the homologous chromosomes need to find their dance partners. They need to find each other. So chromosome set number 1 need to find each other, chromosome set number 2 need to find each other. And that process is known as synapsis. It's not something you have to really refer to right now, but they're finding their dance partner. And when they do, they get so close to each other in their dance partner. So if this is our one from mom, here is our one from dad. They get so close to each other that the spots right there and the spot right here exchange genetic material. So this leads into something that might look like this. Okay, so exchange genetic material. That process here, star highlighted, is called crossing over. This leads into the genetic variability between you, your brothers and sisters, no matter what it is, from the same parents, but the difference is genetic material because this is what's going to go into the egg and sperm when they actually go through crossing over in prophase 1. Once they got done with got their kind of their hug on and they crossed over genetic material, we go into M1, which is metaphase 1, where homologous pairs have found each other, but they now line up side by side instead of vertically. So right now I've got 3 sets of chromosomes. Notice how they're lining themselves up, lining them up next to each other, where in mitosis they're all like this. Okay? Here we notice that they're side by side. Anaphase, anaphase 1, the entire chromosome can be pulled to opposite ends. So here's their centriole, there's their centriole, there's some spindle fibers, chromosomes. So the whole chromosome got pulled apart away, not the sister chromatids. Then we lead into the next one. So telophase 1, where you start to see the guy with each of the chromosomes in here. We could talk about the nucleus maybe reappearing. And then if for just simplicity's sake, we can talk about that it goes and splits these guys into 2 instead of kinesis. Now we need these guys to go through the whole process again. P2, 2, A2, T2, prophase, metaphase, anaphase, and telophase. Now in here, how they line themselves up should be very similar. I'm just going to draw it on one side for right now. They may be all over the place. The nucleus has to dissolve again. Metaphase, now this looks familiar. Just like in mitosis, the sister chromatids are lining themselves up in the middle. Anaphase, pulled apart. Chromatids, that is. And then finally, the telophase. Each one of those cells now has half of what they started with in terms of chromosomes. So if this was the whole thing to do it again, they go through the B2, then 2, A2, T2, and again we'd end with 2 more there. So we end with a total of 4 cells that are having half the number, and then number to it being haploid. Okay. So in terms of major differences that you're going to see in them, again remember that some of the major differences are going to occur in this step in prophase 1, and that being crossing over, and in metaphase 1, the chromosomes align with homologous pair side by side, up and down. Okay. So that just gives you kind of a very brief overview of those. Remember, the whole process of why we're doing this is to get into genetic variability as well as to help maintain our chromosome number. Because if we did not go from that, we went from 46 to 92 to 46. And if this is where egg and sperm was, when sperm met egg, now we'd have a human that would have 92 chromosomes. That's not going to work. And if you think of it, if the 92 were to go through it again, then we'd have more chromosomes be double. And so the process of meiosis not only helps with genetic variability, but it also helps in maintaining our chromosome number. All right, hopefully this helps. Good luck to you, and we will see you tomorrow.